Awesome. Good evening, everybody. Again, I'm Rachel DeRosier, and I'm the program and field trip coordinator, and we are excited for tonight. Thank you so much for joining us. We have Dr. Lucas Majure, who earned his Master of Science degree from Mississippi State University and his PhD from the University of Florida. He previously worked at the Desert Botanical Garden, and then in 2018, he became the curator of the University of Florida Herbarium. His work involves finding and describing new species and patterns of diversity, aka biodiversity exploration. So welcome, Lucas, and share with us what you know about plant exploration and diversity at home and beyond.
Thank you so much. Thank you so much. Lovely introduction. It's very nice to be here with everyone tonight. Really pleased for the invitation. So yeah, so I'll go ahead and let me make sure that everyone can see my screen. I'm sharing it. Does that look good? Okay, I'm just gonna make sure that I've got everything set up and ready to go. Okay, great. So so yeah, as Rachel mentioned, I'm Lucas Majure. So I'm the herbarium curator at the University of Florida Herbarium. So this is within the Florida Museum of Natural History. And I'm going to give you a little bit of background about our herbarium and what we do and that sort of thing and what we do in my lab, and just kind of give you an overview of some of the work that we're doing lately. And then also how that work actually ties into the flora of Florida in numerous ways, right? And so essentially, I'm a plant systematist, so I'm really interested in understanding patterns of biodiversity at different scales. And I'm really interested in understanding groups such as the Cactaceae and the meadow beauties, the Melistomatacea. Those are two groups that I work on quite a lot. Also work a little bit on the grasses and a few other groups here in the Southeast. And so part of what we do in my lab is really try to understand the evolutionary history of different groups of plants, and also and really understand biodiversity regionally. And what that often means is we do a lot of work, say for instance, in and around the Gainesville area and sort of other places throughout the state of Florida, just going in and trying to determine how many species do we have at one location. But in part of doing that, we actually make a lot of collections and we make a lot of herbarium specimens. So, sorry about that, let's go. So I kind of want to introduce herbaria to you guys. I'm sure that most of you have probably seen herbaria, you've probably been in herbaria, but I always like to just sort of reiterate, what is an herbarium? For those of you that might not be totally sure about what an herbarium is, essentially, an herbarium is a library of dried pressed plants, and these plants can these collections that we have sometimes range in order of hundreds of years in age. The oldest herbarium in the world is about 500 years old. Our herbarium was founded in 1891. So it's really the oldest herbarium in the state of Florida, and it's the largest herbarium in the state of Florida. But in terms of what we do with our collection, and I'm giving you guys an idea of what an herbarium specimen might look like here on the left-hand side of the screen. What we do with our collections is essentially these collections form the basis of all the knowledge that we have regarding the species, regarding their distribution. So if any of you have ever used the Florida Plant Atlas online, you will see that oftentimes we have, say, green inlaid counties within the state of Florida. And the reason why those are green is because we have an herbarium specimen for that particular species that you're looking for. So these herbarium specimens actually form the foundation of our knowledge of the flora of any given region. And so in this case, our herbarium actually focuses on the southeastern United States and the Caribbean, but we actually have specimens from across the world from all 7 continents and roughly 500,000 species. There are many herbaria across the world. There are roughly 3,000 active herbaria, at least in the last count that was done in 2017. And this is close to 400 million specimens that have been collected throughout the time that people have been making these types of plant collections across the world. Our herbarium is 91 in the top 100 of these herbaria across the world. So it's nice to be able to say that, you know, we made the top 100 list. It's hard to compete with herbaria such as Paris and New York that have millions and millions of specimens, but we're right at 500,000 collections. So just to give you an idea of some of the collections that we have at the University of Florida Herbarium, roughly 500,000 collections that consist of vascular plants, so that's ferns and flowering plants and gymnosperms, so pines and conifers. We also have lots of specimens of bryophytes, so the mosses and the liverworts, and we have a large collection of lichens as well. So although lichens aren't technically plants, traditionally lichens have been included in herbarium, so we have really large collections of those as well. I just wanted to sort of give you an idea. If you look at the map on the left-hand side of your screen, those are collections that I've made, just those that have been database at least so far. And so a lot of what I do as a plant scientist is I go out and I make collections of plants that I'm studying and general collections of areas where we're interested in understanding what the flora is. And so you can see I've done a lot of collecting in the state of Florida and also in the Caribbean region. Right now in our herbarium, we have about 255,000 of our 500,000 specimens digitized. And so what that means is you can actually go online to our website and you can say, I wonder if they have a specimen of, I don't know, Chionanthus virginicus. And so you can go in, you can type it in, and you can actually see where we have those specimens digitized. You can pull up a nice image of it and you can see the actual location of that specimen, when it was collected, who collected it. And as I was mentioning, these specimens can be used for a variety of different types of projects. And I have just a couple of papers here that we've published relatively recently using collections from our herbarium. So everything from evolutionary studies, ecological studies, describing new species, etc. We use these collections for a lot of different research purposes and citizen science research, many purposes. So how do we get those specimens? How do we make those specimens? Well, it requires lots and lots of field work. I just wanted to show you guys a random assortment of photos of field work that I've done in Cuba, the Dominican Republic, and Peru. I do field work across different parts of the Americas, and both arid parts of the Americas as well as montane tropical parts of the Americas. And so it requires a lot of driving, a lot of time and effort and resources put into going out and collecting these specimens and bringing those back to the herbarium for study. It's part of the job, and it's part of the job that I really love, but it requires a lot of effort. And so oftentimes it's a little more than what people might want to do, but for me, it's really the most fun. This is just a handful of photos to give you guys an idea of, I have this ongoing project to look at the cacti of Cuba. And so this is with my colleague Duniel Barrios from the University of Havana in the National Botanic Garden. And this was part of a field trip that we undertook in 2018. And one of the groups that I work on is Cactaceae. And people often ask me, well, how do you make an herbarium specimen out of a cactus, right? And so this just kind of gives you an idea of how we're doing that. I actually have a recent video that I made. If any of you are really interested in how we do it other than looking at photos, I have a video that kind of explains the process. But this is part of our collection, because I study cacti that we want to diversify in the herbarium. And so I spend a lot of time and effort and resources, both time and financial, going to places and collecting cacti so we can better understand what the species are that occur there, what the overall distributions of those are. And then one major threat to cacti are habitat loss and over collecting for ornamental purposes by amateur cactus collectors. And so one thing we try to do when we're understanding distributions is also understanding population sizes, so we can better predict and build strategies in terms of how to conserve these sometimes very rare species. So all of this information goes into our herbarium, and we use this for downstream work. I was talking about montane tropical habitats. These are some photos taken from the Dominican Republic and Haiti work that I've done there. So there are these really beautiful high montane species of the family Melostomataceae, the meadow beauty family, that we work on quite often in both Cuba and Hispaniola, so in the countries of Haiti and the Dominican Republic. So not only looking for new species to science, but also just trying to understand the evolutionary history of those species that we already are aware of. And so, so we have to, go into relatively remote areas and put a lot of time and effort into generating collections from those areas that we can then study to really carry out our work. So in the end, we have, these are the kind of herbarium collections that we have. So you can see, cacti, although they're really spiny, they're oftentimes super succulent, they actually make really nice herbarium specimens. And part of this work too is really aesthetic, right? But it's really like making art. When you go out and you collect a plant specimen, there's all of that information tied to the specimen that can be used for, literally an infinite number of years for research down the road and for many other reasons. But it's also creating a little bit of art. And that's something that I really love when I'm going out and I'm making herbarium collections. I'm also sort of tapping into that artistic side. So it's really a lot of fun, especially to see these finished products. So each one of these herbarium specimens is going to have my collection number and collection date. It's going to have the locality where it was collected, maybe who I collected with, GPS coordinates. It's going to have a little bit of information about the ecology. And so these collections, we try to make them as rich as possible so many different people can have access to them and so they can use them. So that's sort of a long and short of what we do in my lab. I wanted to introduce some of the students and postdocs in my laboratory. We have a really great group here at the University of Florida and within the herbarium. And I have students working on the families Cactaceae, the families Melostomataceae, and from a number of different countries, including the US. And so, so we really have a great group of young budding scientists right now that I'm training to work and do work on not only the floristics and understanding biodiversity at local scales, but understanding evolutionary relationships and really understanding, okay, when we go out and say, that's Smilax bononox, well, how do I know that that's Smilax bononox? How do I know that that's Smilax temnoides, for instance? I actually have a grad student right now, Thomas Murphy, who's working on Neotropical Smilax. A lot of people don't like greenbriers, but it turns out there's about 100 species in the American tropics of Smilax, and they're not all very easy to tell apart. And there are probably a lot of species that have gone undetected. So my grad student, Thomas, is actually currently working on that. And other students are working on other projects. And so it's a really great group of people. And it's a lot of fun getting out into the field with them, training them, and collecting plants and really trying to understand the patterns of biodiversity and the overall numbers of species in many of these groups that we work with. So I wanted to sort of take you from this field work that I'm talking about and take you through the process of how we generate data and what we do with those data. And so this is one group of cacti that I work on. They're super diverse in Western North America. They're called the choyas. You might have heard of them as the jumping choyas, or a jumping cactus. If anyone's ever been to the desert Southwest and the Sonoran Desert or Mojave Desert or Chihuahuan Desert, you will have seen species of the genus Cylindropuntia. Those are the choyas. And so this is a paper that we published in 2019, but it essentially just shows the evolutionary history, or hypothesis of the evolutionary history of this group of plants. And but what a lot of people don't realize that goes into, say, an evolutionary analysis of an evolutionary history of a group of plants like this is you really need to know the plants, first of all, before you do any kind of lab work. And you have to do a lot of field work so you can understand the plants that you're actually going to be grinding up and sequencing DNA of later on. And so what I'm showing you here is, so first of all, we go out into the field, we collect these plants, we take lots of data on those plants, and then we have an herbarium specimen that represents a plant that might be included in our downstream analysis. And then we generate, in this case, DNA sequence data that we would then compare across species to understand species relationships, and maybe even to make determinations about, okay, well, I think I can tell this species apart from that species, but what do I know about their relationships? Are they really closely related? Are they not closely related? Oftentimes, and I know that you personally have dealt with situations where you go, is that this species or that species, or are they even different species? And so part of our job is actually to figure that out and tease those patterns apart. And so that's what we do with a lot of this DNA sequence data and morphological data that we collect on the species. Also ecological data. You know, sometimes it's pretty easy to tell a species apart based on where it's growing. You might think of Quercus hemispherica and Quercus larifolia in Florida. Oftentimes I know which one I have because of what habitat I'm standing in, right? And so those different types of data can be incorporated into these types of analyses to help us make determinations about what we should be calling a species. So I want to jump into the Caribbean just for a moment. And you might go, well, I thought we were going to talk about Florida plants and Florida biodiversity, but actually to understand parts of the biodiversity in Florida and the flora of Florida, we have to think outside of the box a little bit, because if you think about the flora of Florida, it's a fascinating area. I'm actually in southern Florida right now. I'm in Miami. And if you work your way down to the Florida Keys, for instance, you're going to see a lot of plants that are shared with both the Bahamas, Eastern Cuba, and sometimes Hispaniola, Puerto Rico, and the Lesser Antilles. You might also see plants that you would find commonly in, say, tropical Central America. But then in other parts of Florida, you're going to find plants that are actually more closely related to dry, arid areas of Western North America. Or you might even find plants in northern Florida that are more closely related to plants in the Appalachian Mountains. And so the flora of Florida is a fascinating really mix and hodgepodge of different floras that have come together to generate the types of communities that we see across the state. So it's just absolutely fascinating. I've been for many years fascinated with the family Cactaceae, and this is just sort of an evolutionary tree of the family Cactaceae. And what I want you to pay attention to more than anything are these stars that I have on the tree, and those stars represent the number of times that this family Cactaceae has made it into the Caribbean islands. And so essentially what we've had over and over and over again from continental areas such as Mexico, Central America, and then South America are these long distance dispersal events into the islands. And more groups that we work with are showing this very same pattern. So it's a super interesting pattern that we see. But so what does that mean from the standpoint of Cactaceae in Florida? Well, there's this really interesting genus called Consolea. It's a genus that I've been working on for a number of years now. It's a close relative of the prickly pears, the genus Opuntia. And we've been doing lots of work with this genus on the islands of Hispaniola, Jamaica, Cuba, Puerto Rico. And one thing that we've also been trying to do is work out the overall evolutionary history and that story or a hypothesis of the evolutionary history of this group. You can see the size of some of these plants is absolutely massive, and it makes you think of the giant Galapagos prickly pears. But this is actually in the Caribbean islands, and so they've essentially evolved this large tree-like form separately from those giant species that you see on the Galapagos Islands. But there might have been some of the same selective pressures to do so. So it's really fascinating. Well, we've recently generated an evolutionary tree for this group. And one of the species that I'm really interested in is this species from southern Florida, Consolea coralicola or coralicola. So this is often called the Florida semaphore cactus. One thing that's automatically noticeable about this species is it's very morphologically similar to this widespread species Consolea milspaughii. So Consolea milspaughii, I actually took this photo in eastern Cuba, and the one up top is actually one that's in propagation. But Consolea milspagi is found in the Cayman Islands, it's found in eastern Cuba, and it's found in the Bahamas. And it's interesting, if you track the distribution of the species, and then if you look at the evolutionary history of this small group, which I have outlined here on the left in blue, you can see that within milspagi, Corallicola sort of sits there, right? So Corallicola is super closely related to milspagi, and it probably evolved outside of Consolea milspagi. So really, Consolea corallicola represents essentially a northern distribution of this more widespread Caribbean species. And so what this tells us is that this species that we have in southern Florida, in the lower Florida Keys, is a species derived from the Caribbean flora. So this is an aspect of our flora that's derived from the Caribbean. And so what's super interesting about that is that gives us this sort of a view into the past and helps us understand how the flora of Florida has evolved through time and how we have developed the flora in different parts of the state. What about if we look at other prickly pears? So this is a group of prickly pears that I worked on for my PhD. And there's some really fascinating plants that occur in especially peninsular central Florida. So you can see these really large upright shrubs here in the middle and on the top right-hand side of your screen. But we also have smaller species like this at the top left-hand side and the the bottom right-hand side of the screen. And so I was really puzzling over these as I was working on this group for my PhD. And part of what I wanted to understand was, okay, what should we call a species in this group? And then where did this group come from? And there's actually some interesting populations in the Florida Keys that for a long time were considered part of this larger Caribbean group. And so that was something I wanted to try to tease apart. So in doing this, I looked really, really closely across a very wide distribution. Many of these species occur over a pretty wide distribution. And one thing I wanted to do was also look at the number of chromosomes in this group, because this is a group where there's lots of hybridization. Sometimes with hybridization, genomes double. And when a genome doubles, that means your number of chromosomes doubles. And so you can actually count the number of chromosomes and make decisions about how or whether or not the genomes have doubled. And that gives you a hint as to whether or not there's been any hybridization in this group in the past. So I took a really close look at this group, and in doing that, I found that it was pretty interesting that it seemed that the putative ancestral populations of this really large group that occurred over most of the United States, those ancestral populations seem to be restricted to the southeastern United States and then to part of southern Texas and then eastern New Mexico. So that's really strange. So it seems like these populations that potentially could have hybridized, leading to this other large group of populations that extends all the way to Canada, were actually derived from these southeastern populations. And what that kind of tells us is that there's probably in the evolutionary history of this group, because over evolutionary time, if you think about climate change during the Pleistocene, you had a lot of an increase in the overall excuse me, go back there. So you had an overall rising of seas, and then you had lowering of seas, and that kind of happened on and off throughout the Pleistocene. And with populations being able to come together and then being pulled apart and coming together and being pulled apart, you can actually get hybridization scenarios where you could develop this type of pattern. And then with sort of that ending and us being in this interglacial event now, you can actually get populations that are restricted to the southeast and then parts of the southwest. Well, so in looking closely at some of these taxa in southern Florida that were supposed to be part of this Caribbean group, I realized very quickly that this material we used to call Opuntia triacantha in the Florida Keys was actually not the Caribbean Opuntia triacantha at all. So when I actually ran a phylogenetic analysis to understand the evolutionary history of this group, I saw that our Florida Keys material actually fell out with the Eastern North American group of species. But that being said, we have a number of other species that occur within Florida that are actually much more closely related to species outside of Eastern North America, such as species from Central Mexico and Northern Mexico, and even the Western United States. So there's this pattern sort of back and forth of relationships of cacti in the state of Florida. So, and taking a look at this pattern of cacti in the Florida Keys, I noticed that there were differences in chromosome numbers of some of these, and the overall way that they looked, their morphology was quite different. So I was comparing our material from the Florida Keys to material from the Caribbean. And it was very clear pretty much from the onset that the morphology was really different. And then, of course, looking at the evolutionary history of this group, I see that, okay, this material from the Florida Keys that we've been calling Puncha tricantha is actually not tricantha at all. It comes out way down here in another part of the evolutionary tree. And then this other material from Cuba, which we thought was tricantha, is totally different. There was this other material we had called a Puncha cubensis in the past, or another taxonomist had done that. And I realized that's just a really strange hybrid between this material from Southern Florida and another species. So the type of work that we're doing in my lab is directly related to understanding the evolution of the flora of Florida. And so sometimes you have to really dig into a group like this using lots of different types of data. In this case, DNA sequence data, chromosome number, the morphology, so overall how the plant looks, to really determine whether or not something is what we think it is, right? And so oftentimes we'll see that even in the flora of Florida, which we think is super well understood and very well studied, we have situations like this where actually taxonomists in the past have just gotten it wrong. And sometimes it deserves a little more comprehensive research in developing really robust data sets to test some of those hypotheses. And so I would say, in general, we understand the flora of Florida quite well, but there's still a lot of work that needs to be done. So just sort of reiterating that we were using the wrong names for these for a period of, probably 30, 40 years. One other group that I work on that I wanted to just sort of outline really quickly to you guys is the family Melostomataceae. So I'm sure that probably all of you have seen the Meadow Beauties, and there's actually a chapter of FNPS, which is the Rexia chapter. And so there's this is also a really fascinating story, given that the vast majority of Melostomataceae are tropical. They're both New World and Old World tropical, with the vast majority of species from this family, about 3,500 of the over 5,000 species, being found in the American tropics. Well, it turns out the Meadow Beauties, this fascinating group of about 11 species of these small herbs within the family Melostomataceae, they occur from essentially Puerto Rico all the way up to Canada. But the southeastern United States is the absolute center of diversity for this group. And within the Southeast, really Florida has probably some of the highest numbers of species of Rexia than anywhere else. So we have a couple of species within the state that are actually threatened because they're known from just a handful of populations. And there's some cool disjunction patterns that we see as well. For instance, this yellow-flowered Rexia lutea is essentially disjunct between the Panhandle and the Atlantic seaboard. So it's a really, really fascinating group of plants to study. But one thing that is often interests me is that Rexia cubensis, which was described from Cuba, as you might imagine, it occurs in one part of Cuba, it occurs in some of the savannas of Hispaniola, and then it occurs in Puerto Rico, and then it's relatively widespread in the southeastern United States. But so with the Consolea before, I was talking about this movement from the Caribbean north, right? And so our populations of this consolea actually came from the Caribbean. Well, it turns out there are certain groups like the meadow beauties that potentially evolved or at least diversified mostly in the Southeast, and then they dispersed south. So the components of Rexia, so Rexia cubensis that occurs in the Caribbean region, actually came from the southeastern United States. And this is one of these patterns that's super fascinating to me, because you get this sort of patterns of plants going back and forth, some moving more north, some going south. And then one really interesting facet of this and trying to figure out how this happened is thinking about dispersal ability. And so these form these capsule fruit that have these tiny little seeds. You can see an example of one of these seeds up at the top left of this one photographic plate. And so some of these are head scratchers. You think, well, how did these species move from one place to another? In many situations, birds are some of the primary distributors of or dispersers of some of the fruits and seeds of these groups, but not necessarily so in the case of Rexia. And so this is one of those situations where it's a really interesting phenomenon and series of disjunct populations that we see in this group. And we're trying to wrap our heads around this and really generate hypotheses as to how we might have how this type of pattern might have evolved. And we actually not only see this in Rexia, we see it in a lot of other groups as well. One of those groups is actually the genus Dicanthelium. So I know I threw up a grass and people may or may not get excited about this. This is actually one of my favorite group of grasses. And they're roughly 70 to, I would say, between 70 and 100 species of this grass. This is another grass that's essentially endemic to the Americas. There are 4 species on Hawaii, but otherwise it's endemic to the Americas. And the vast majority of species are found in Eastern North America. And then within Eastern North America, the Southeastern United States is that absolute hotspot of diversity for this group. And so this is a group that we've been working on for a number of years now. We're actually getting ready to publish a phylogeny showing the evolutionary history and species relationships of many of the Eastern North American species in this genus. So this is one of the old panicoid or panic grasses. It's a genus that's been segregated from the genus Panicum now for quite some time. I really think they make beautiful ornamental species. I actually have quite a few planted at my house in Gainesville, Florida. I think they're absolutely gorgeous plants, but they're also fascinating from an evolutionary perspective and from a taxonomic point of view, because they're incredibly difficult to determine at the species level oftentimes. And one thing we're finding with this group of grasses is that sometimes species that morphotypes, I should say, so what we call a species, but it has a different look across different parts of its distribution. We normally have considered many of those the same species, but what we're finding is actually a lot of those are cryptic species. So they're really hard to tell apart, but they actually should be recognized as separate species based on what we're seeing from their evolutionary relationship. So it's a super fascinating group of grasses to work on. They're really super diverse in pine flatwoods and moist sandy soils. That's where the diversity just goes crazy in this group. But you also find them in rich mesic forests and xeric hammocks and hydric hammocks. You can find them in a number of different habitat types. This is actually another group that has moved north to south. So we find a lot of these species in the Caribbean region and high elevation montane areas, which kind of act like the temperate zone. And that's where we find a lot of species of this genus Dicanthelium. So this is one we're actively working on in my lab, and this directly correlates with our understanding of the flora of Florida. And it actually turns out, even based on some of the recent treatments of this group for the flora of Florida, they're still a little bit outdated, because we're going to be recognizing a number of other species in this group that you could consider cryptic species that haven't necessarily been recognized previously. And so there's still a lot of ongoing work in this group to better understand them, but we're making a dent there. So I'm really excited about getting this work out and the implications for conservation and our understanding of the flora of Florida and the southeastern United States. One other project that I wanted to bring up tonight, just because we're talking about the flora of Florida, and I think that it's a group that oftentimes goes way undernoticed and underappreciated. And it's a group that I've always, had sort of a soft spot in my heart for, and it's one that I've started working on quite a bit. And those are the bryophytes. One thing that we've been doing lately, just on the University of Florida campus and some of the University of Florida properties like Ordway Swisher Preserve, is we're actually collecting all the mosses and all the liverworts and hornworts in those areas. And we've been generating these, what we call barcode data sets for those. So we have some genetic data associated with each one of those collections, and we can, along with identifications, we can use this genetic data to try to determine the number of species and the overall diversity of bryophytes within an area. And it's been an absolute, incredibly fun project, and I'm learning a lot from it. And I'm actually finally learning how to identify a lot of the mosses and liverworts that we have locally in the state of Florida. And so I'm really excited about pushing this project forward. I have one of my graduate students and a former graduate student that have been helping tremendously on this project. And so they're also being trained in learning how to identify these mosses. And I think this is going to give us a really nice tool in terms of, oftentimes we go into areas and do floristic surveys and analyses of the vegetation, but we oftentimes leave out the bryophytes because they're so tricky. But I think with these sort of new methods and tools we're developing, we'll actually be able to incorporate those in a meaningful way and really understand how much diversity we have of the mosses in these areas. And one thing we're finding, even within the Gainesville area, Alachua County has an incredible number of collections, but we're actually uncovering things there that haven't been collected, say, in 100 years. From the University of Florida campus, I've recently made some collections of mosses that have just gone unnoticed for a really, really long time. And so we're definitely filling in the holes and the gaps of our knowledge. So in our lab, another thing that I was mentioning that commonly happens is when we're out doing field work, then we're working in the herbarium, we come across these species that turn out to be new to science. And I just wanted to outline a couple of these that we've found recently. These are not in Florida. These are actually on Hispaniola, both the Dominican Republic and Haiti. And this is a group of melastomes that I worked with for my postdoc. I was recently out with colleagues in the Dominican Republic, and we came across this material, and I automatically realized that it was a new species. And then I compared it with other species that had already been described, and it was quite clear. Then putting this into phylogeny and understanding the evolutionary history and species relationships, very clearly a new species to science. And so this is something that we're in the process of describing at the moment. We also just recently described this new species to science from the Tree of Heaven family. This is the Symerubaceae. So we have a native species in Symerubaceae to South Florida, Symeruba glauca, but there's also an invasive Ailanthus altissima, the Tree of Heaven, that you can find especially in parts of northern Florida. But this is a different group altogether that is most diverse in arid areas across the Americas. So it's in southern South America, it's in the western North American deserts, but it happens to be most diverse in the tropical dry forest of the Caribbean region. But it has gone undernoticed and understudied in the tropical dry forest of Hispaniola. And so we've been working on this group quite a bit in the last few years and have described this one new species, and now we're working on another new species. So this came about because we were actually doing a floristic study in a mountain range in southern Dominican Republic, and we ran across this material. And the point I would like to make with this is that there are many areas, even in the state of Florida, where we're missing lots and lots of data in terms of what we know about the regional floristic diversity. And so in working out floristic patterns by making collections and identifying those collections and just getting out and seeing the plants, we can oftentimes find that we're underestimating biodiversity, even in a relatively well-studied area. And actually, over the past number of years, there have been a number of new species described from the state of Florida. So even right here in our own backyard, where we think we have it all figured out, that's not always the case. So it's just a sober reminder that we need to be out there looking at plants and collecting plants and making sure that we're training students to do the same thing. And so that's really where my heart lies. That's what I really love to do. And so I just kind of want to end here with some perspectives in terms of the work that I do and what I think is super important. So this floristic herbarium taxonomic work that we do is really needed more than ever before. Some of the species that we have that even within the state of Florida are totally lost. So we realize that there's a lot of urbanization that's happening in Florida. And we need to really be getting out into areas and collecting and making sure that we're understanding the flora before potentially, and I'm in Miami, I'm in South Florida, where, really less than 1 or 2% of the rocky pinelands still exist. And so this is one of those types of areas that's especially under threat. I feel like we really need to be comprehensively training students and others so they can learn how to make collections, identify plants, just getting people out into the forest and into the pinelands and anywhere else, and having them really figure out how to tell one plant from another and sort of, pry us away from our computer screens. I try to do that as much as possible, just so I can learn more and so I can help others learn as well. And then incorporating some of these other methods that we use is also super important. But really, we really need to be training not only students, but young ones as well, right? Our kids and everyone else to get out and enjoy and really value plants. We need to be doing lots of work in these poorly known groups of plants, but also poorly understood areas. So there are a number of counties in Florida, for instance, let's see, Hendry County, Osceola County, that have really amazing natural areas. So, for instance, we were recently in the DeLuca Preserve, which is a recently acquired property from University of Florida, and doing a little bit of work out there, trying to understand the overall diversity of that area. Those places that have very poor collections, we're often going to find new county records and things like that. So certain species that we might have considered threatened or endangered, we might find new populations of those that can kind of help us fill in the gaps. In my capacity, I'm really interested in student training and getting students out there, getting dirty, getting their hands dirty, looking at plants and understanding the plants and making collections and understanding how herbaria work and why they're important. And so that's really, really where my heart lies. And that's what I feel, that's what I'm super comfortable and happy doing. So kind of just want to leave you guys with that perspective. Some of what I've shown you today was derived from funding from, of course, Florida Museum of Natural History, National Science Foundation, National Geographic Society. But we also do have an herbarium fund that we do get donations to that help us go out and do a lot of this work, or some of this work that I've been showing you, especially when it goes to training students. So recently our work down at DeLuca Preserve was funded by our herbarium fund. That's what my lab's here for. That's what I'm here for, is getting people out there and learning about plants and loving plants and trying to make sense of nature. So hopefully it's right at 7:51. I'm always a bit long-winded, so hopefully
Yeah, that was fantastic. All right, so now at this time, we are opening it up for any questions somebody might have regarding the presentation. I do have two comments.
Okay.
Stacy Perry wrote triangle versus circles.
Yeah.
I don't know what that means.
Oh, triangles versus circles. Maybe, well, I don't know. Maybe she was referring to the tree diagrams that I was showing.
At the very beginning. It was towards the beginning.
Let's see.
It looked like the distribution map.
Oh, okay. Yeah, absolutely. Let's go back to that distribution map. Let's see. Perhaps this distribution map here. So here, yeah, this part, I kind of went through this relatively quickly, but essentially what I'm showing here is that the black circles are what we call diploids. So they have 22 chromosomes. Those with white circles have 44 chromosomes, and then those with gray circles have 66 chromosomes. And so this is showing kind of an incremental increase in the genome duplication in this group. What's interesting about genome duplication that we commonly call polyploidy is that it's thought that with an increased number of chromosomes and increased number of DNA, plants can actually really withstand more adverse conditions. And so oftentimes we might see plants that have duplicated their genome in much what we would consider at least harsher conditions. And so in this case, you see a lot of these polyploids way up in the cold Midwest, or on really hot, dry areas along the Gulf Coast, whereas you see the diploids in perhaps more favorable conditions. And so that's one of the interpretations of sometimes why you get this whole genome duplication in plants.
Allison Bishop, I think it was a little bit after this, she posted beautiful specimens. I think it's just a couple slides.
Oh, thank you. Let's see, I can go back and, I really, I love making specimens, actually. So I'm teaching a tropical botany course at the moment at David Fairchild's old property and at Fairchild Tropical Botanic Garden. And actually we had I did a workshop on Saturday just showing the students how to make specimens, because it's really kind of a lost art, and it's something that people might even perceive as archaic, but actually having a specimen makes all the science that we do repeatable. And without having a specimen, without saying, so I could crush up leaves all day long and pull out DNA and I could sequence those, but unless somebody has a way to verify what I've done, then it's kind of pointless, right? And so by having this physical specimen, someone can say, well, I wonder if Lucas actually sequenced what he said he sequenced. They can actually go back to the specimen and they can confirm whether or not I was, doing what I said I did. And so it's a way to make our science repeatable, which is super important.
Pam Calendar, she's saying awesome regarding, I think it's regarding the presentation. Tom Heitzman, he he says, great talk. Which species of the, oh gosh, I'm going to butcher this, Dichthelium?
Dichthelium, yeah.
Dichthelium, do you think has potential landscape potential?
Oh, yeah. So I think it would depend on where you live in Florida. But for instance, if you lived in, so let's say you're in Central Florida, and you have, it would also depend on, are you dealing with, moist soils or dry soils? But for instance, there's a really nice woodland species, Dichthelium commutatum, that's a nice, robust, kind of shiny leaves. It really grows throughout the year. I think that has a lot of really nice landscape potential. There's another large species that occurs in mesic forests in northern Florida called Dichthelium boesii. And I actually have that planted in my front yard, and I think it looks beautiful. It has large sort of velvety leaves and makes a really, really nice ornamental.
What was the species on that one again, Lucas?
Boesii, Bosscii. Yeah, it's
great.
a really nice species.
other one that you were saying?
The other one was commutatum, Dicynthelium commutatum. Yeah, that's, yeah, yeah. The problem with some of these Dicynthelium is they don't all have good common names, and so I think we should probably just generate some really good common names for some of these that don't have them, so we can have a better handle on them.
We have a couple more questions. I wonder if this is Anna Madden. It says, I wonder if rafting from Amazonia during hurricane events long ago is a piece of the regional diversity.
So that's actually a really good point regarding the distribution of some of these species that we see in the Caribbean. Rafting and meteorological events like hurricanes, we know that those are actually moving around some of these cacti that we work on. And I've actually seen this firsthand after Hurricane Katrina on the Gulf Coast of Mississippi. Just a week or so after Hurricane Katrina, I was doing some work down on the Gulf Coast, looking at all the devastation from the hurricane. But one thing I noticed was there were prickly pear pads all over the beaches because there were some outlying barrier islands right off the coast, 10, 14 miles off the coast. And numerous parts of those populations had been ripped out by the hurricane and they had been deposited on the beach. A year or so later, I go back and they're actually growing there. And so what that means is that, and we knew this, but they're tolerant to salt water. And then they can actually form new viable populations after being moved, a tremendous distance over, a certain number of days in salt water. And so I think the very same thing has happened numerous times in some of these taxa that have moved throughout the Caribbean. I think it makes perfect sense.
Allison Bishop says, I love tasting Smilax and a great variety of flavors.
Oh, yeah.
Opportunity to prepare specimens, and it's quite challenging to do it right, let alone attractive.
Yes. Yeah, I actually I joke with my student Thomas all the time, because I work with cacti and all these other thorny things, but Smilax are tough, right? So they have all the prickles on their stems and they're really tough to work with. And then it's really good to kind of get bits and pieces of the rhizome in the specimen as well. So they're not easy to make good specimens out of. And so I leave a lot of that up to him. but if you take the time and sort of have the perseverance, you can make some really nice specimens out of Smilax.
And then she also says, when you are collecting from rare habitats,
Mm-hmm.
you take part of the plant so that the species isn't as affected.
Oh, absolutely. Absolutely. One thing that we always have are ethical considerations. And so if we're dealing with plants that are rare, endangered, et cetera, we're super careful that we're not endangering any part of the population, the reproductive potential of that population, because otherwise we would be undercutting what we're actually trying to do in the end, right? Mm-hmm. And so we would be really undermining conservation efforts for a species like that. So we're very careful, very cognizant of what we're collecting, when we're collecting it. And always, I could have had an entire presentation about permitting and appropriate permitting and those sorts of things. That's something that we pay a lot of attention to and make sure that we're doing appropriately. Absolutely.
Well, I think that concludes our questions, and we're good on time too. So everything works out there. Okay. I thank you very much. It was a pleasure to listen to you, and I can just feel your passion and your excitement about plants. That's lovely to see from other people. So thank you very much for being here. Thank you for all of your information. And yeah, welcome back anytime.
No, absolutely. And thank you so very much for having me. It was an absolute pleasure. And whenever anyone's ready to come up to Gainesville and come visit us, just let me know. Send me an email or give me a call and come on up. We'd be happy to have you.
All right. Yeah, we might actually be in touch.
Yeah, absolutely.
Yeah. Stuff in the works. Okay, so and as far as the group goes, don't forget we have that field trip coming up for this Saturday. And I hope that you all get to enjoy your early mornings in the summer and the late evenings with your son. And we'll see you guys again in September when we start back up. And we'll be sending out information through the summer regarding our program so everybody can get organized. I know that's appreciated. And let's see. Oh, and in the chat, I did go ahead and send that Colony Cove link, but I'm going to go ahead and also make sure we get that emailed out to everybody. I looked into it and I think I might go there. That sounds like a great, looks like I think it's a Saturday volunteer opportunity. And also we had Sean shared his BioBlitz information. It's in the chat. Looks like we've got some BioBlitzes coming up in May to May 23rd, May 27th, June 3rd, and June 15th. Everybody's just very happy. Thank you, thank you, thank you. And oh yes, there's a field trip form link from Rachel. Thank you, Rachel. And there is still opening, so go ahead and sign up. And Tom says, can you help? Okay, what were we saying there, Tom? About
ID some witchgrass, which is the dicantheliums.
Sure.
They're hard to tell apart.
They are quite challenging.
I guess the best way is where they're found, their habitat, maybe.
Habitat's really helpful, and then spikelet size, pubescence is super helpful. The length of the ligule at the apex of the petiole or the leaf sheath, those are all really good characters.
Great. Thank you.
Absolutely.
Okay, so I guess we are good. We will be in touch. Have a wonderful summer, everybody, and I look forward to seeing you again in September. Okay, guys.
Thank
Happy
you.
summer.
Great evening. Thank you.
Thanks, Sharon.